Delighted to be welcoming to our virtual stage Eran Segal. He's at the Weizmann Institute of Science, and he's going to be joining us today to talk about personalized medicine using clinical and microbiome data. Eran, thank you so much for joining us.
Thank you. Let me share my screen. Okay, can you see my screen?
We can, yes. So I'm going to drop hand straight over to you. Thank you so much, Eran.
Okay, thank you. So my lab has been working in the area of microbiome for quite some time. The first major project that we published several years ago on personalized nutrition. We assembled a cohort of 1,000 people. We profiled their gut bacteria. We connected all of them to continuous glucose monitors. And for 1 week, they were asked to log everything they eat, so that at the end of the week, we had from all the 1,000 participants, 50,000 meals with blood sugar responses. And the key findings of that study were that people have very personalized and different blood glucose responses, even when they eat exactly the same meal, as exemplified here in the response of people to bread. You can see similar responses of the same person on different days to bread, but very different responses across people. We were able then to develop an algorithm that predicted with good accuracy the personalized meal responses of these participants using clinical data and gut microbiome data. And in a small cohort, we showed that we can take people who are pre-diabetic, who normally have these spikes in glucose levels, and we can fully balance their glucose levels by giving them a diet which is tailored by the algorithm with exactly the same amount of calories as the diet that spikes their glucose levels. And then in a follow-up study that we just recently published in Diabetes Care, we did a randomized clinical trial, 200 people with prediabetes that we randomized into either our standard of care diet or into our algorithm diet, and mainly looked at changes in glucose levels after half a year of intervention. And what we found, this is anecdotally one participant where you can see prior to the intervention, 1 month of continuous glucose monitoring data showing spikes in glucose levels that were fully normalized on the first month of the intervention, and also in the entire 6 months of the intervention that's shown here. And that's, of course, anecdotally just one participant. But when we summarize the main statistical findings of the study, we saw that after half a year, this is the 6 months point here in hemoglobin A1C, which is the gold standard and average measure of glucose levels in the past 3 months, we saw a reduction in the standard of care diet, but a reversion back to baseline. Whereas in our algorithm diet, we saw double the effect, which was also sustained for then the next 6 months of follow-up. So for us, this was very satisfying, completing a journey of 10 years from an idea to development of an algorithm to a randomized clinical trial to show the efficacy of this for treating people with prediabetes. And so when we finished this study, we asked whether we can use this approach of population cohorts to go more broadly beyond just nutrition and study more broadly health and disease. And that led us to developing what we call the Human Genotype Project, where really the goal is to try and change this curve. This curve shows you the cost for developing new drugs, which has really skyrocketed. We're now developing new drugs is over $1 billion per drug. It seems that in the past decade, there's been a plateau in this graph, which some people attribute to the use of genetic data in the drug development process. And because genetics is just one component, it's also a fixed component that does not take into account the well-being and status of the person and the environment. We believe that by using deep phenotyping data about people, we can maybe hopefully reverse this and lower the cost of new drug development. And so we formed a first milestone of reaching 10,000 people. We completed 8,000 of these people who we profile. We also follow them longitudinally. And what happens is that some people remain in their healthy baseline status, some progress towards disease and some develop disease. And together with that, we've also profiled some specific disease cohorts in oncology and cardiovascular disease that we compare to. This project has been going on for 3 years. We have 15,000 people who registered, and as I mentioned, we already profiled 8,000 of them. And the broader vision is to expand this to a very large cohort of up to 100,000 people and profile additional disease. At the base of this is technology that we've been developing for many years in the lab to really lower the cost and increase the throughput by which we can do this profiling. What is this profiling that I'm talking about? When people come to a visit at the Weizmann Institute, they undergo clinical and molecular testing. The clinical includes measurement of the cardiovascular system using both ECG and ABI. We connect them to continuous glucose monitors. We get their medical records, so all the diagnoses they have. We use ultrasound to look at fat in their liver, a DEXA machine to look at full body composition and bone density. We use sleep monitors, which give us information about the sleep quality of people during the night, many analog signals of blood saturation and other signals. We record their diet. We biobank samples that we can apply future assays to. We record their voice. We use wearables. And on the molecular side, we do full genome sequencing of the host genetics. We do sequencing from the blood to look at RNA gene expression. We are planning to do proteomics. We're doing microbiome profiling from the gut and of the oral microbiome, and we're using metabolomics to profile thousands of metabolites in a person. And then we have a new assay to assess the immune system of a person by synthesizing hundreds of thousands of antigens, and then with sequencing, measuring what antibodies recognize out of these hundreds of thousands of antigens. So in a single assay, we can detect the entire infectious history of a person, for example. So you can see we profile many different systems of the body with essentially almost all of the molecular measurements that we know how to do. And the question is, okay, what do we do with all of this data, and what can we learn from it? So because the time is short and this is a microbiome symposium, I want to focus on what we're finding in that area. And so in that area, what a cohort of thousands of people allows us to do is to really develop new techniques for analyzing the microbiome. And this is going beyond just the composition of bacteria. So once we take these metagenomic samples and we map them to a reference genome database, we also now record the exact genetic variation at every single position across each bacteria. And we can then associate this genetic variation at the single nucleotide level in specific bacteria with various traits of the host, like an example here, associating genetic variation in the species with a BMI of the host. And when we do that, it turns out that we are finding very significant signals. So this is a key result of this analysis. Every dot here represents one position in an individual bacteria where genetic variation was significantly associated, even after Bonferroni correction, with a trait, in this case with BMI. The y-axis shows the p-value of the association. You can see it's very significant. The x-axis shows the size of the effect, so how many points of BMI statistically is explained by every SNP here. And you can see that some of these associations are striking. They're explaining 1 or even 2 points of BMI. This can be 5 or 10 kilograms of body weight, 10 or 20 pounds of body weight. And then when we do a deep dive into the actual findings, we see that in some cases, the SNPs actually go together. They're correlated with each other. So here's an example of one species of bacteria, where on the x-axis are the SNPs that came out as significant. The x-axis, that's the x-axis. The y-axis is the number of people, 1,300 in this case were analyzed. And you can see that the SNPs kind of cluster and they form 2 different bacterial strains. And if you compare individuals that have here the dominant strain, the major strain, that's close to 1,000 people, and they have a BMI that's lower by 2.3 points, again, a lot, can be 10 kilograms of body weight, as compared to individuals, the 300 individuals that harbor the minor strain. So this is a very dramatic, very significant finding, and we believe that this could be potentially a bacteria that would be beneficial to give to people who are overweight, to give them the strain, in this case, the strain that's present in the majority of the population. Here's an example of another similar strain that, similar picture of a different strain that we find. In this case, a strain that's explaining 1 point of BMI. And again, you can see this clustering between the major and the minor alleles. And here's another such example, this time explaining 1.3 points of BMI. This strain is actually a major producer of butyrate, a short-chain fatty acid that's been shown to have clinical utility. And so we're now developing a pipeline where what you just saw is a discovery pipeline, a computational discovery pipeline that works at the level of SNPs. It identifies bacterial strains that associate with our traits of interest. And we are now working to isolate the relevant strains, grow bacterial consortium from them, and then go into clinical trials to test their efficacy for the various traits that hopefully they can help with. So I want to end in the last couple of minutes that I have showing you another place where we've done an intervention based on the microbiome. This is in atopic dermatitis, where we tried to do a fecal microbiome transplant. This is a skin disease. It's very prevalent, affects about 20% of children, 10% of the adults. And we did an FMT trial where each patient was his own control. Each patient received initially placebo capsules, so capsules that do not contain any fecal material, separated by 2 weeks each for 2 dosings. And then they were given 4 dosings of fecal material from a healthy donor. And they were then followed up for several weeks later. And I'll first show you the clinical outcome of these results. So you can see that after the placebo, there's clear symptoms of the disease. But then after 2 dosings of the FMT, we can see clearance of these symptoms. Here's another patient with clear symptoms that are then cleared even many weeks after the last FMT. And when our clinicians quantify this, they see about a 70% on average reduction in clinical symptoms. This is a proof of principle trial on just 9 patients, but 100% of them responded. Some responded better than others, but all of them responded and had a 70% on average reduction in clinical symptoms. When we analyze the gut bacteria, we see very clearly transfer of bacteria from the donors. Again, we're doing this analysis on the SNP level. And what you're seeing here is every row is a different species. The box plots show the distance between the genome of the species when you compare unrelated individuals. So you can see there's a typical distance of 1%, sometimes 0.1% across the genome between unrelated individuals. And the dots show the same comparison, but between the donor and the patient. You can see that at baseline and after the placebo, the dots are at similar distances to where you find unrelated individuals, because of course, there's still no relationship between the donor and the patient. But after we do the FMT, we see a lot of dots move all the way to the left, indicating essentially no distance between the donor and the patient, indicating a transfer of bacteria from the donor to the patient. And my final slide is showing what happens to the microbiome globally. You can see in ellipses here, samples from the donors clustering together, samples from the patients are somewhere in the space. After the placebo, there's not a lot of change, but after we do the FMT, we see that there's a major shift in the microbiome of the patient, and he becomes now not identical, but much more similar to his donor. And this happens for almost all of the patients in this trial. So with that, I'll just put up my acknowledgement slides for collaborators first, which we had many on all these projects, and then members of my lab who drove this work. And with that, I will end, and thank you.
Thank you so much, Eran. We've had a few questions in already. If you do have questions for Eran, please do use the Q&A box at the bottom to put some of those questions in. We don't have long to put some questions to you, but we've got a couple that I'm just going to put forward now. The first one I want to ask you is about fecal transplants, actually. We've had several in about that. So do you think oral input of probiotics or even fecal matter could be as efficient as the proper protocol for fecal transplantation?
Could it be, sorry, could it be the proper protocol? What?
Could it be as efficient as the other protocols referencing?
So I think in the end, fecal transplants are a good way to do a proof of principle study. But then later on, when we want to develop therapeutics, that's probably not going to be the approach, and we want to reduce it to a set of bacteria that will contribute, and that's what we're after. What I mentioned in both the trials that look at various phenotypes from our large cohort, and also in this atopic dermatitis study. Now we're trying to reduce the effect to a number of species that we want to isolate and then test as a therapeutic.
Sure. We've got a question to ask you about the atopic dermatitis patients. Were they, you showed up to sort of 33 weeks. So was that shift stable long term? Did you follow up after that or anything?
So that varied between patients.
Sure.
And in some patients, it was actually stable for a long time, even for many weeks after. So in some patients, we actually, yes, we did see colonization for a long time. In others, we actually saw colonization for shorter time periods. And in some cases, we even gave them additional doses and saw, again, better clinical effects.
Thank you so much. I've got an interesting question here on what you think about the relevancy and accuracy of things like metagenome assembled genomes, MAGs. Is there anything you can comment on that?
Yeah, so these MAGs, metagenome assembled genomes, are probably of lower quality than isolates. They're lower quality of what we can assemble with long reads. We actually have work that we just submitted to review on working from tens of thousands of samples and assembling both MAGs, but also assembling with long reads, which I think will be more accurate. And I think these genomes are good to work with because that's what we have now, but we need to improve them by methods such as isolation, which is the best probably, and then long reads, which we also found can assemble complete genomes and with good accuracy.
One final question, if I can just squeeze it in. The question was, what methods are you using for your SMP detection?
Yeah, so these are methods that we developed. Basically, you map the reads, you record the variation that you see at each position in each person in each sample, and we quantify the major allele, and we look at the major allele frequency in each sample, and that's what we use to do all the correlations that I presented.
Great, thank you. We are pretty much out of time now, I'm afraid, Eran. If there's one or two things that you just would like people to take away, or sort of a quick summary, what would they be? If I can put you for sort of a quick, concise 30 seconds.
Yeah, so I think we are finding that in the field of microbiome, one has to assemble large cohorts, as people did in genetics, and only once we reach such cohorts, we'll be able to find more robust results, as I think we are now beginning to find, and as just really follows from the field of genetics. So I think that we should strive for large cohorts that are phenotyped in a very broad fashion to look at multiple different phenotypes, and I think that will be a good way to move forward in the field, and will allow us also to develop these more novel and in-depth and higher resolution methodologies, like the type of SNP approaches I showed.
Thank you so much, Eran. Absolute pleasure to have you with us. I know that you're a very busy man, and we appreciate you taking the time to present with us today. And an
Thank you.
absolutely fascinating presentation. So a huge thank you to Eran Segal for